Thanks so much for the kind introduction and welcome everyone. It's a really great pleasure to be able to speak here today, especially following Ed. It's always a pleasure to listen to this whole discovery story, which is really one of those big detective stories I feel in environmental chemistry and toxicology. So always glad to listen to you. So today I want to talk a little bit about those species differences in toxicity that we observe. The question, I hope I will be answer a couple of those in the talk today, and really focus on some species of fishes of commercial, cultural, and ecological importance. And now I'm at the University of Saskatchewan, smack dab in the middle of the country basically, and so we're really interested in the relevance of this compound to inland fisheries and species of fishes of importance to inland bodies of water as well, right? Because initially there was quite a bit of debate about, is this isolated to coho salmon or is this a broader issue? So I hope I'll be able to show you that there is actually a much broader issue behind this compound. Actually, before I get started, I want to make sure to also acknowledge that this is a whole team effort. You see a lot of names here on that first slide, a lot of collaborators across the University of Saskatchewan Toxicology Centre, as well as the University of Lethbridge in Alberta, as well as DFO in Winnipeg, and a couple other collaborators that we have in the room here as well. So really proud to be able to talk about this today. The background I will keep very short because Ed alluded to that already. You basically got all that information straight from the horse's mouth, and so I will not go into much detail here, but then really go into those initial objectives that we focused on, really trying to characterize acute and sublethal toxicities of 6-ppd quinone to a number of different species. Really also trying to dive into the mechanisms of toxicity. What is going on and what is the difference between those species of fishes? Why are some so sensitive and others are not? And then last, actually, to probably answer that question around the metabolic differences, we looked into the biotransformation products of 6-ppd quinone, basically the ways in which fish are trying to detoxify the chemical, or perhaps bioactivate. There's still a big question mark about that, right? So we'll look into that a bit more. So you heard this just before. There has been this mass mortality events, really unsightly and really tragic to see. Basically, we invest all this money, all this effort into trying to renaturalize those streams, and then something like this happens. So it was really great, and Ed is probably too humble to really be bragging about this as much as he could, because this is a fairly marvelous feat. Very rarely you're able to take an observation of population and ecosystem relevance and boil it down to that one single chemical or that one single group of chemicals, right? Ed mentioned it, it's probably one of the most toxic chemicals that we know today. And this little speck of red chemical here is driving all this toxicity in the nanogram per liter range. If you were to look for a good analogy for that, it's basically 1 grain of salt in an Olympic-sized swimming pool, right? That's not a lot of chemical that's killing fish acutely within very short time frames, right? So this really, when we saw this paper come out, we already knew we had to do something and we had to get involved in this topic to some extent. We already had some collaborations with City of Saskatoon around stormwater quality, not looking in particular at 6-ppd quinone, but in general. And we had those samples floating around. So we dropped a quick email to Ed and Jen Yu and said, hey, could you share that standard with us so that we can start looking for it? And there you go, 6-ppd quinone in our stormwater runoff, in our snowmelt, actually at fairly sizable concentrations. So we knew now, actually, we have detections in Saskatoon as well. There's no coho salmon in the South Saskatchewan River, but still, seems to be an interesting and important topic for us to look into, right? So we did that. At the time when we started our studies, coho salmon were really that only species that was known to be sensitive, right? Now with that revised LC50, 95 nanograms per liter, very, very toxic chemical. All other species of fishes, and that included some of our own data, we looked into fathead minnows, we looked into zebrafish, these kinds of things. None of those species were sensitive. A couple other papers came out of Norway, out of Japan, and they all showed the same thing. None of these cyprinid fish species, like the zebrafish or like the medaka, were sensitive to 6-ppd quinone at really high concentrations, hundreds of nanograms per liter. Nothing happened to those fish, right? So the big question was, okay, is this an artifact? Is this something unique about coho? What is it that drives this? And so there clearly was a need to better understand how widespread sensitivity to 6-ppd quinone was and what the mechanisms are that drive this. So this was really our goal to dive deeper into this. So we were quite lucky. We had a couple of different species of fish in our aquatic toxicology research facility at the University of Saskatchewan at the time this came out. So we could basically start right away. We included a number of fish species in that we had floating around. Some collaborators at the University of Lethbridge then also jumped onto this with brook trout that they had. And so we were able to fairly quickly look into this. This all grew into a much bigger list of fish species that we've tested to date. Like I mentioned, it included the rainbow trout, one of those very standard model fish species that are fairly important in toxicity testing, but also important recreationally and commercially. This included the brook trout. We also had some Arctic char and some white sturgeon in the facility from other studies that were left over and were just waiting for a good use. So we were able to do this basically over a Christmas holiday. A little bit of a weekend science project actually at the time. And so we went into this not expecting much to happen after nothing happened to our fathead minnows. And we actually started this just before a holiday party in my lab. And then I get frantic text messages and phone calls. Oh, the fish are all dying. What do we do? And so this was certainly exciting in good ways, but also in bad ways, right? So this was with the rainbow trout at the time. Nothing really happened to the white sturgeon or the Arctic char. We then also included brown trout and bull trout into this, west slope cutthroat trout. So the list of species really grew quite big in collaboration with a number of different labs. So just to give you a little bit of an overview of our methods, we basically exposed those fish for 12 to 96 hours, depending on their sensitivity, as subadults or juveniles. So this was basically with the older life stages that we looked at before they turned sexually mature. But then we also did experiments with early life stages, starting from hatch all the way to swim up. And that was 28 days for rainbow trout and 45 days with lake trout. So quite a bit of work. Katie Roberts actually is in the audience here. We'll talk about that in a bit more detail tomorrow. So I'll just give you the highlights today. We conducted all of these experiments as static renewal experiments. So that means every 24 hours, someone had to go into the lab and change 70% of the water. So that's certainly handling a lot of water and a lot of manual work, which kudos to the students for pulling all this off. The test concentrations we picked, they were basically informed by limited number of replicate pilot tests where we just picked a high concentration of 6-ppd quinone and to see whether or not they would fall into that sensitive or not so sensitive category. And then after that, we picked species-specific concentration ranges for the actual tests. We did confirm the concentrations that our fish were exposed to using similar types of methodologies that Ed was mentioning earlier, liquid chromatography with high-resolution mass spectrometry, really trying to track what happens with a 6-ppd quinone over time. Does it decrease in concentration, and if so, how much, right? Just so that we get accurate LC50 values. Now, I already foreshadowed some of those results in Arctic char, brown trout, bull trout, west slope cutthroat trout, and white sturgeon. Even at the highest, and now these are measured concentrations that we exposed them to, of greater than or equal to 13 micrograms per liter, very high, much higher than what you find in the environment. Nothing happened. Those fish were just fine and happily swimming in that 6-ppd quinone solution, right? So those are clearly insensitive to those exposure concentrations. Now, brook trout, rainbow trout, and lake trout were quite different. They showed significant mortalities with those median lethal concentrations, or LC50s, of 0.33 to about 1 microgram per liter. So you see that here in this range, basically. They all fall within that range between very little, a couple nanograms per liter, up to 1 microgram per liter. And now the other thing you will notice here, the subadult life stages here, they really group more on the right. So they're less sensitive compared to the early life stages, right? So that means either the early life stages right after or around hatch, up to exposure of the fry of those species, they are much more sensitive compared to the older life stages, right? About a factor of 2 or so. So that's something to keep in mind as well. Life stage dependent sensitivity, Ed alluded to this as well for the coho salmon. Certainly something that, that happens here in these species as well. And like I mentioned, Katie will talk about this a bit more in her talk tomorrow. Now, what's also interesting is that temporal aspect behind all of this, right? So on the left here, you see the brook trout, then rainbow trout, then lake trout, time on the x-axis, and then the probability of survival depending on those exposure levels. In this case here, there are actually nominal concentrations over time. You see in brook trout, a very rapid drop-off here within a couple of hours in the highest 2 concentrations. Those fish all died within 6 hours, right? Very similar here with the lake trout fry, but the rainbow trout, it took much longer, 12 hours, 24 hours for those higher exposure concentrations to actually act on those fish, right? So there are some differences in the kinetic response to exposure with this chemical, how quickly they respond. And that certainly is important when you think about the flashiness of 6-ppd quinone, the rapid increase of concentration with a rainfall event, and how quickly it then decreases again, right? So definitely something to keep in mind. The other thing, again, differences in those life stages, right? So on this left-hand side here, we see the rainbow trout early life stages. Actually, over this entire period of 28 days, it takes them a long time to actually show mortalities, right? So it's quite important to keep that in mind. While the same age at the end of this, basically 28 days plus a bit, you expose them to the same concentrations of 6-ppd quinone, and you get this rapid mortality within a couple of hours, right? So again, there's a bit of a difference. It might be driven by the bioavailability of 6-ppd quinone to those larval life stages. It might be less available than to the fry life stage here. And at the same time, they are exposed over those long periods of time. So if there is some biochemical driver to this, perhaps some enzyme is induced or upregulated in those fish, and it might be slightly protective for them, right? So definitely something we need to look into here. Now, I want to show you some quick videos here. Those are those subadult brook trout on the left, and then some fry here on the right. I believe those are rainbow trout, Katie. Yes. You see the exact same behavior that Ed showed earlier for the coho salmon in the field and in the lab. The spiraling motion, this gasping at the surface, right? And this is a very reproducible etiology here between all those different species. So very interesting to see. So now if we put all of this together, our own studies here in blue, as well as literature values in orange, you really see there's basically 2 camps here among the fish species. All the sensitive ones are on the left here, all the insensitive ones are on the right here, and we lump them all together as being greater than 12.7 because that was the lowest high concentration that was reported as measured. All the others might have exposed to higher concentrations, but nothing happened, right? So it's really basically those 2 distinct groups. Now, what's interesting about this is that even closely related species like the brook trout and the Arctic trout here, both are salvelinus species, could be either very sensitive or entirely insensitive, right? So phylogeny doesn't really seem to be a good descriptor of what's going on in terms of sensitivity. And that was really something that piqued our interest because, you know, this would be your first assumption. They're closely related, so they would be also sensitive or insensitive. But apparently that was not the case. Now, interestingly as well, to date, only salmonids have been shown to be sensitive to 6-ppd quinone. I'm not aware of any other study. Maybe there's some knowledge out here. But so far, only salmonids would be in this sensitive group down here. There's no cyprinids in there, nothing else that has been studied to date, right? As well, we did a bit of research with invertebrate species, and it seems that at those environmentally relevant concentrations, nothing is really happening, again, in any group outside of the salmonids, right? So quite interesting. Now, I did overlay this here on the phylogenetic tree, right? Again, you see all the Oncorhynchus species up here, all the Salvelinus species down here, and then there's Samo, like the Atlantic salmon and the brown trout down here. All of those are insensitive, but then you have a really mixed bag. Some Salvelinus species are sensitive, others are insensitive. Some Oncorhynchus species are sensitive, others are insensitive, right? So again, your phylogenetic tree doesn't really predict what's going on in terms of responses. This is actually an interesting one as well because we focused a lot on those acute lethalities, but we tend to forget about those sublethal effects, right? Things that you can observe in fish before they actually start dying. And so there are things like this here after 15 days, these are all rainbow trout. There you see a yolk sac where there shouldn't be one. So that's a yolk sac retention. That should be gone by that time after 15 days, but it's still there. So it clearly indicates something's going on with energy metabolism there in those fish species, right? You also see these kinds of edemas that Ed alluded to that you actually see quite often after exposure to stormwater. That is driven by 6-ppd quinone. And then those spinal deformities here, right? Really strong curvature up to this kind of corkscrew here, combined with a jaw deformity, combined with a yolk sac edema. So there's really a triple threat here basically for this larva. And then this here on the right really puzzled us. Up here you see a tail fin of a healthy rainbow trout at 15 days. Nice and clear, well-developed tail fin. And this is after exposure to 6-ppd quinone. So you see all this blood pooling in between those blood vessels where it shouldn't be pooling, right? This should be flowing right back. We've sent this picture around to a number of fish pathologists, and they all tell us they have never seen anything like it. Something similar after viral infections, but then it's more splotchy. It's not as uniform, right? So definitely something that piqued our interest. And now this is actually something you don't only see in the rainbow trout, but you also see in the lake trout. So again, very, very reproducible, right? And again, Katie will talk about that a bit more tomorrow if you're interested. Now, all of this gave us a lot of initial evidence to start looking more into mechanisms, right? We knew something was going on with the development of the tail fin. We knew something was going on with energy metabolism, as indicated by that yolk sac resorption. There was also some indications of changes in how glucose was metabolized in those sensitive fish species. And we also knew something was going on with that gasping on the surface, the spiraling motion. It all seemed to be almost like a suffocation from within in those fish species, right? So all of those were pointers, and we wanted to look into this in a bit more detail. Ideally, what we always try to do is establish these adverse outcome pathways or toxicity pathways by which you can predict from a first interaction of a toxicant with a macromolecule in a biological system what will be triggered in terms of cellular responses, organ responses, and then organismic responses all the way to the population level, right? So this will be our ideal case where you have evidence supporting all of these points. Probably not quite there yet, but we have supporting evidence along the way. And so I want to talk about that a bit more today. So first, a PhD student in our lab, Hannah Mahoney, she was interested in that energy metabolism question, right? But in order to not have to use live fish all the time, she decided she wanted to go with permanent cell lines. And we had some rainbow trout cell lines, rainbow trout liver cells, RT-LW1, and gill cell lines, RT-GILW1 in the lab at our fingertips. So we decided to expose them to 6-ppd quinone. Fairly high concentration if you look at this here. This is really just to get an idea of the mechanism rather than environmentally relevant levels, right? And expose them for 24 hours. After that time, we looked into 3 different endpoints. We looked at cytotoxicity. Was that 6-ppd quinone able to kill our cells? We looked at oxygen consumption rate. Did it change how the cells were able to respire and to use oxygen for fueling their metabolism? And last, we also looked at the biotransformation of 6-ppd quinone. Does that system turn the 6-ppd quinone into something else in the process of being exposed to it, right? And again, we did that using our high-resolution mass spec here in our lab. Now, I don't show the cytotoxicity results. You would see that only in the gill cells at the highest concentration, at 80 micrograms per liter, there was a slight decrease in viability. Not really meaningful. But at the same time, you look up here, those are our gill cells, and down here are the liver cells. There is a significant increase in oxygen consumption rate only in the gill cells and not in the liver cells, right? So there clearly seemed to be a difference between those two cell lines. Now, the big question was, what does that mean? Higher oxygen consumption rate, we actually followed up on that, could mean that your mitochondrial respiration, that means the powerhouses of your cells are basically impaired in how they do their job, right? It's currently unclear how 6-PPD quinone does that. It seems that it's through a non-traditional pathway, but there's extra research that is needed to really confirm what is going on there. But again, that really stark difference between the gill cells and the liver cells, that was something that was noteworthy. So we looked into whether or not 6-PPD quinone was actually transformed into something else by those cells. You look at the gill cells up here. There's only 6-PPD quinone present in those, both in the exposure media as well as in the cells that were exposed to it, right? You look at your liver cells, there's very little 6-PPD quinone left, and it's almost all converted into a hydroxylated metabolite. So your organism actually attaches a little OH group to the 6-PPD quinone, and by that, either bioactivates it into something more toxic or detoxifies it into something that is less toxic. And since we didn't observe any cytotoxicity in the liver cells, nor any effects on the oxygen consumption rate, but did observe that in our gill cells, we concluded from this, at least in a preliminary fashion, that this hydroxy metabolite is actually a detoxified form of 6-PPD quinone. For the time being, that's a good hypothesis to work with. Other groups out there, I will just put that out as a disclaimer, think actually that a hydroxylation might be leading to an active metabolite, right? So this is something of active ongoing research at this point. Now we also looked into cardiorespiratory physiology, right? We saw this gasping, we saw this spiraling motion, we saw an increase in oxygen consumption rate. So we wanted to make sure that we actually understand if the same happens in our subadult fish here. And so we did that with rainbow trout and Arctic char to really contrast a sensitive and a non-sensitive species. We measured oxygen consumption over time. Again, we collected water samples for chemistry to make sure that we know what's going on. We also collected traces of ECGs, basically. Like you were to go to your doctor's checkup and get your heart checked out, we did the same with our fish. We also then used Doppler ultrasound to do the same thing, to determine the volume of blood that your fish can pump through the body per time unit, right? All of this to really get a good feel of what's going on with our fish. And this is all work by another master's student in our lab, Summer Selinger, who has won many awards for this kind of presentation, so really great work. And I only want to summarize a couple of the findings here. So long story short, there was no difference in oxygen consumption in those fish exposed to 6-PPD quinone or the control. That was a bit puzzling to us, so we didn't quite understand what's going on here. But mind you, those were all sublethal concentrations, right? You need to have a live fish in your tank in order to be able to observe how much it breathes. So perhaps we were just below those threshold concentrations, right? What we did find, though, we measured a couple of blood parameters, and this is interesting. Up here, you see the Arctic char, the tolerant species, and down here, the rainbow trout, our sensitive species. Nothing really happened in our Arctic char at all between the controls and our treated groups here in red. But when you look at the oxygenated hemoglobin, there's a significant decrease there, right? You look at the deoxyhemoglobin, so hemoglobin without oxygen bound to it, you see a significant increase. So it clearly shows that our fish are transporting less oxygen with their blood through their bodies, right? Why that is the case, we don't know at this point, but certainly an interesting observation. And that is before the fish actually die, right? This one here is also very interesting. Methemoglobin is a chemically modified form of hemoglobin, which is unable to effectively transport oxygen. And so an increase in that will actually impair your ability, again, to transport oxygen through your body, right? So again, something very concerning. If you had elevated methemoglobin levels during your annual checkup, then your doctor would certainly be very concerned about you, right? So something to keep in mind here as well. Now, I mentioned we want to really try to unravel what's going on in terms of the mechanisms. What are those individual steps that lead to all of these effects and all these symptoms that we can observe? So we use those early life stage fish here again, in this case, rainbow trout. And those are the same fish that we exposed for 28 days and that I showed you pictures of before and so on. But what we also did was after 4 days, we subsampled some of those fish larvae and we froze them, we extracted the RNA and basically sequenced the entirety of all those RNA molecules. So basically all those little blueprints of genetic material that are then turned into proteins in the body and sequenced them. And with that method, you can basically determine transcriptome-wide if you have responses. Do those genes go up? Do those genes go down, right? How does the fish respond to exposure with 6-ppd quinone? If you put it all together later on, through bioinformatics tools, you can actually put together a very compelling picture of what's going on with your fish, right? Much more detailed, actually, than any annual checkup at your doctor if you were to go. This is work, again, by another master's student, Jeannie Lin here, who is working in Markus Hecker's lab at the University of Saskatchewan. And so I'll try to walk you through this. It sometimes gets a bit overwhelming, but this volcano plot here basically plots how much a gene is changed and whether or not it's statistically significant on those two axes here. All those red ones here are significantly upregulated compared to the control group, and all those green ones here are significantly downregulated compared to the control, right? Overall, it's a total number of 1,400 dysregulated genes. So 1,400 different potential mechanisms that we need to look at in this organism to understand what's going on. That's a fairly large number for any chemical. And so those of you who have done these things, you oftentimes end up with a couple dozen, maybe a couple hundreds, but 1,400, that's a large number of dysregulated genes, right? If you plot them according to their similarity, all those individual fish here, in terms of their similarity between one another, you can definitely see that there's some clustering going on here. So this is the high exposure group, and they are dissimilar in their reaction from the control group, which is right in here. And then you see all these other exposure groups, they get more and more similar as the concentration decreases, right? So clearly a concentration dependence in this. Now, for every single one of those genes, those 1,400 dysregulated genes, we can try and fit a concentration response curve, just like we did with the acute mortalities that we observed, right? Concentration on the x-axis versus the difference in expression on the y. And then you can try and fit a curve through that, and hopefully you go somewhere. We were able to do that for 689 of those dysregulated genes, right? So again, a pretty large number, almost 700 genes where you can fit a model. What you can then do based on this is you can find the concentration here, the vertical lines, the red ones that you see, and derive what we call a benchmark dose. So that's the dose of 6-ppd quinone at which that particular gene is now dysregulated significantly, right? And again, you do that for all the genes in your list, for the 689 here, and for 668 genes, we could find a concentration-dependent difference in expression after exposure to 6-ppd quinone, right? Again, very large number. Now, you can do all sorts of interesting statistical things with that. The first thing is you can actually plot each individual gene here according to their benchmark dose on the x-axis and the number of genes for which that happens on the y-axis, right? So the most interesting point on that curve is this peak here. This peak is basically the concentration at which most of the genes that were significantly dysregulated were dysregulated, right? So that basically means this is where most of the biology actually happens. And you look at this concentration here, maximum first peak, 0.35 micrograms per liter. And that's awfully close to our apical benchmark dose here, so when the fish die at 0.47. But mind you, 0.47 is determined after 28 days of exposure, and this benchmark dose here is determined after only 4 days of exposure, right? So we can look at our fish larvae after 4 days of exposure and predict with some certainty how sensitive they would be down the road. So it's almost like an early warning system, right? So very powerful. In terms of what kinds of pathways are dysregulated, we see there's a lot of things like innate immunity, immune system response, inflammatory response, cell differentiation and development. So that's interesting for tail fin pathologies, right? As well as cell death. It's basically individual cells being triggered to die from this exposure. And again, this is very interesting because the study that came out by Justin Greer on early life stage coho salmon found very similar things, right? So again, there's a little bit of reproducibility across the different species. So very interesting to observe. Okay, so you can put all of this together in a pathway model basically and try to come up with what all of those potential effects might be. So again, this is tentative, not complete by any means, but maybe a good start. So we see this uncoupling of mitochondrial respiration, which changes how the fish is actually cycling oxygen through the body. It decreases the available ATP pool to fuel any of the activities like cell proliferation, so you get a decrease in growth, right? You get also an increase in reactive oxygen species, increases oxidative damage, triggering cell death, ultimately leading to death of your fish, right? And there's other things like chronic inflammation, which might increase these kinds of things, edema, triggering death again, and so on, right? There's probably a number of other effects that we haven't described yet where we need to look into. There's probably also other initiating events that we haven't discovered yet. There's a lot of studies that went into potential neurotoxicity as well. So a lot of things we need to keep in mind, and we don't want to discredit as not happening or not occurring. We just haven't observed them in our studies, right? And for all of that, you can actually predict them with our molecular models, right? So it's quite interesting to see that. Now, last objective, and just very quickly, I want to summarize some of the work that we're doing around the biotransformation of 6-PPD quinone. There's a number of other papers that are currently coming out. Another preprint will be submitted probably tomorrow, and then there's another one coming out in 2 weeks or so. So a lot of action currently in this space. But we already published this one here, and again, this was a nice collaboration, again, here now with collaborators from Washington State University. Jen McIntyre contributed some efforts to this, as well as friends from Huntsman Marine Science Center, Ben DeJordan here, and Danielle Philibert helped out with this as well. So what did we do? From all of those studies that we did before with subadult and juvenile fish species, as well as a couple new ones, we collected gallbladder bile, right? So when you metabolize in your liver, a lot of those products of that biotransformation are shuttled into the gallbladder bile, and then from that they go into your gut, right? That happens in our system and many other systems. So it's very well known. It's a very nice matrix to collect in order to characterize chemically. So we diluted those samples in methanol, spun them down in order to get rid of sediments and any precipitation products, and then again ran them on our LC Orbitrap high-resolution mass spec. And this now used a very similar workflow to the one that has been applied initially to actually find that co-occurrence of tire chemicals with ERMs in coho salmon, right? Very similar workflow. So we tried to vary in an unbiased fashion, basically, look for those biotransformation products. And this is work by yet another master's student, David Montgomery, who just graduated a couple weeks back. And you've seen this hydroxy 6-PPD quinone before. This is what we described in our cell cultures. So that was no surprise that we found that one again. And you can see from left to right, from sensitive to insensitive species, you got an increase in the concentration of that, right? So that was interesting for us to see. At the same time, usually what happens to those hydroxylated products in our bodies, and this is commonly known for polycyclic aromatic hydrocarbons and other chemicals, after that has happened, our bodies attach a little glucuronide, like a sugar group, to it, to make it even more water soluble to facilitate getting rid of it even more, right? And this one we observed here too. I believe this was one of the first studies to show that in fish. And again, you get a concentration dependent increase from the insensitive fish species to the sensitive fish species. Sorry, the other way around, from the sensitive to the insensitive. And so I must send a little disclaimer with that. Obviously, those ones here, because they were so sensitive, were exposed to much lower concentrations compared to the ones on the right. But even if you detrend that, you will still see that difference in abundance here. So this suggests to us that toxicity or the absence of toxicity might actually be related to the ability of our fish to get rid of 6-ppd quinone from the body quicker, right? And now some of those biotransformation enzymes we're currently looking into, which ones are responsible for that. Not quite there yet to really release this, but it looks like the baseline expression might be different across those different fish species. They have very similar enzymes across all these salmonids, but their baseline expression in an unexposed fish might be different, right? Which might trigger all those species differences in sensitivity. So overall, I guess I was able to show that select salmonids are highly sensitive to 6-ppd quinone at those levels we frequently measure in the environment, right? This is not isolated to coastal populations. This also has a lot of inland relevance. And in particular, when you look at lake trout, this is something that certainly in the Great Lakes we are very concerned about, right? There's unique behavioral responses which do suggest to us that something is going on with respiration, perhaps those neurological effects as well. Our in vitro studies do suggest that toxicokinetics might actually play a role here in those species differences in sensitivity. And that the gills might perhaps really be one of those important target tissues that we need to focus on, right? So in addition to the acute toxicity, the fact that we were able to observe early life stage toxicity really shifts the focus now. Ed mentioned it. It's now important to not only look at fall when it rains and the fish are trying to move up the stream, but also in the spring, when we have those massive numbers of hatchlings or early life stage fish trying to migrate down again, right? So definitely something to keep in mind and not always just focus on those acute toxicity events. All right. With that, again, I would like to thank our funders. Much of the research has been supported by Department of Fisheries and Oceans here in Canada, as well as through Canada Research Chairs, Natural Science and Engineering Research Council of Canada, Innovation, Canada Foundation for Innovation, CFI, and Western Economic Diversification. And I want to thank you for your attention. Thanks.